Somebody confirm that you can hear me.
Yeah, we can hear you.
Great, thank you. Let me shut down my email. All right, let me go to the grade book just for a little bit and then I can talk about what I've got graded. I know I got the last lecture worksheet and the lab worksheet graded, but I don't remember what they were, so let me look here. Worksheet for lecture 2 has been graded. It looks like almost everybody got full credit on that too. Good job. And worksheet for lab 2 for the first grading has been done. Surprisingly, I got worksheet for lab 1 graded. It's kind of rare that I get the second grading for the early labs done, but for lab 1, I have the second grading done. I have not, don't ask me why, I did not do the grading for lab 00. For some reason, I started on lab 1 for the second grading. So if you did not get 100% in the worksheet for lab 2 on the first grading, remember you can turn it in by this Saturday, 11:59 p.m. for a second grading, and that will be for full credit. So in the labs, you get to submit the labs twice to get your final grade. Any question about any of that? The reason is I consider the lab a learning exercise, so you're getting two shots at it. In the lecture worksheet, remember you only have one or at most two questions, and you only get one shot at it. So if you do not know the answer, you should either one, look up the answer, or two, wait for another student to take that question, and then you take another question later down, the next question, when it's your turn if you don't know the answer and you know a later question. Any questions about any of that? All right, let me get out of the grade book and then I'll share my screen again. All right, you should be able to see my screen now. Not much to see there, but actually, let me go back to that. I need to do something. Where is that? After I'm finished with the lesson, I do display the PowerPoint slides. So I will make lesson 1 available to you. And if I don't have that done a day later, you can send me an email as a reminder. Another thing to do is when I share my screen, I don't see very much that what's going on. Um, like the only way I can tell that I'm actually recording this is that little red button right there, little red dot there. And if I don't share the screen, I can then see that it is not being recorded or it is being recorded. Where the heck is that? There it is there, right there too. Of course, you can't see that because I'm not sharing my screen now. So when I'm sharing my screen, I don't see all the information. And if you see that I'm not recording the lesson, please let me know because I do want to record these lessons and then post them on my YouTube website to make them available to students who cannot attend the live Zoom session. Any question about any of that? And as a reminder, I do not use the chat I do not use emoticons. I do not use the hand up symbol or anything like that. The reason is when I'm sharing my screen, I can't see that information. The chat is most of the time hidden from me, as well as the other screens. So they said when I first was taking Zoom that you really need two instructors in a class to handle all of the features for Zoom. And guess what? You've only got one instructor for this class, so I'm just not going to use the chat. If you have a question, you need to either state it in class, meaning unmute your mic and then just interrupt me and ask your question, or you could always wait until I stop this for a breath of air and then ask the question, or you can email me, in which case you'll get the answer later. All right, let's turn to the syllabus. Today is October 8th, and let me blow that up a little bit so I can see it better. Um, Today we're supposed to be finishing chapter 2. I don't know if we'll get there, but we're supposed to. And then on Wednesday, we have our first real quiz. For those of you who did not do the practice quiz, you should make sure that Respondus Lockdown Browser is working on your system. I don't know how you can do that, but you do need it working before you can take the quizzes. And if there's a student who recently joined the class, you do need to download Respondus Lockdown Browser to take the quiz. Any question about any of that? Quiz 1 will be covering Chapter 1 and Chapter 10, the very start of Chapter 2, meaning what we covered last week, not what we're covering today, and then Labs 0, 1, and 2. Any question about any of that? Would
Yeah,
you,
I do. Oh, sorry. Do you mind me asking if it is open note or is it closed note?
the labs are open book, open note. However, it is a timed exam, so you will only have time to look up one or two answers. And if you look up more answers, you will run out of time on the quiz, and then you will get those questions wrong. So you do need to know the answers before you take the quiz. Is that clear?
Totally. Yeah, thanks.
Okay. Like I said, you do have enough time to look up one or two answers, but beyond that, you do not have enough time. Um,
I had a quick question, if that's
yeah.
okay. Is it all multiple choice, fill in the blank, or there's some essay questions that I need to prepare for?
It's all multiple choice for quizzes 1, 2, 3, 4, and 5. The final does have two parts, where final B, which is most of the questions, are multiple choice. And then there is a final A with maybe 4 or 5 questions. And this is not a timed uh, exam, meaning final A. Um, and you can obviously look up the answers too, but it is fill in the blank essay questions as well as I don't know, like a worksheet. And when you're doing the unknown project, it will be similar to trying to figure out what your unknown bacteria is. I realize you don't know what the unknown project is because I haven't talked about it yet, but you will have a bunch of tests, the results to the different tests, and then you'll have a table to look up which species belongs with that result, and then you have to determine which species it is. And it's very similar to the unknown project. So that's it for the final, but for all the quizzes, they're all multiple choice. There was too much cheating on the essay questions, and so I gave up on them.
How much time should we set aside for the quiz tomorrow?
I give you 1 minute for each question, and then maybe it depends how many questions there are, but about 5 extra minutes for the quiz. So 1 minute plus for each question, and then about let's say 3 to 5 extra minutes, depending on how many questions there are.
Sounds good.
Okay. And it states on the quiz how long it is, so you can go take a look at that. And once you start the quiz, you have to finish it because the timer starts running. And once the timer runs out, you can no longer answer questions in the quiz. Now that we talked about steroids and sterols, is that correct? So slide 30. All right, didn't hear anything, so I'm going to assume that's correct. Our next topic is proteins. Proteins are essential in cell structure and function. Proteins may be many different things. They can be structural proteins, such as proteins found in bone, muscle, and hair. They can be enzymes, which speed up chemical reactions. They can be transport proteins, which move chemicals across the cell membrane or another organelle membrane. They can be regulatory proteins, which regulate body functions, like a hormone, like children produce a growth hormone to help them grow. And there are proteins responsible for movement. The reason why my arm is moving is because of proteins, and that's why we walk and everything else when we're moving. There are proteins that are responsible for some bacterial toxins, and a toxin is something which is poisonous to us. And then there are some proteins, antibodies, which help defend the body against a foreign invader, especially a foreign microbial invader. And that just tells you the large number of different functions that proteins can perform in the body. Any question about any of that? Proteins do consist of a monomer. The monomer subunits are called amino acids, and you simply hook the amino acids together to make the protein. The generalized amino acid structure is shown here. Let me load this up a little. They all have a central carbon and then a hydrogen attached to that carbon. On one side of the amino acid, they have the amino group, that's NH2. On the other side of the carbon, they have the carboxyl group, and that's COOH, with that first O having a double bond to the carbon. And then they have a fourth group. Remember, carbon binds to 4 different atoms. A fourth group that we're calling R. R can stand for essentially anything. It's the rest of the amino acid. There are 20 different amino acids in humans, and most organisms have about 20. Most actually do have 20, but there are a few that have, I think, a few more, 21 to 23. I think the highest number of amino acids in a bacteria is 23. The R stands for the side group that makes that amino acid, meaning there's different side groups here. This side group there is the R, and this would be tyrosine, which has a OH group and a carbon 6 chain plus the carbon methyl group there. The R group determines which amino acid it is. And like I said, there's 20 different amino acids in humans. That means we have 20 different R groups in humans. Any question about any of that? Now, you don't need to know the different R groups that make the 20 different amino acids, so don't memorize this table. I'm just showing you some of the different R groups. Blow that up a little bit. The simplest R group is hydrogen, which is in glycine. I'm not seeing the color there. I think that one's the R group right here, and that's the hydrogen attached to the central carbon. So glycine, all it has for the R group is an H. The next most simple would be alanine. It has a methyl group for the R. And then there can be branched methyl groups or chained mesh methyl groups. And all of these, including this one, the R group is non-polar. So all of these amino acids like being around each other. They also like being in the membrane because the lipid bilayer is non-polar. And so is that one, non-polar. But there are some R groups which are polar, such as tyrosine. This part of the molecule is non-polar, which is most of it, but this OH attached to it makes this R group at least partially polar. Any question about that? And then the NH group on these two NH groups on this R group makes it polar. This one is polar too from that NH group, and that one is nonpolar. Any question about any of that? So the important thing to remember is that proteins have non-polar R groups to their amino acids. So they can be partially non-polar a protein, but then all proteins are always polar. And that's because the amino group, where do I have that shown? The amino group here and the carboxyl group are polar. And some of the R groups are polar as well. Many of the R groups are non-polar, in which case a protein is both polar and non-polar, because there will be many amino acids in a protein. So there will be some non-polar regions in the protein. Now, for a protein, which is in the cytoplasm or in any other place of the cell, which is around other polar molecules, such as water, the non-polar regions of the protein will form the three-dimensional structure of the protein, and the non-polar regions will be tucked on the inside, and then the polar regions will be on the outside of the protein. Conversely, a protein around a lot of lipids or nonpolar regions, such as a protein spanning the cell membrane, which is mostly nonpolar, obviously it depends on which side of the membrane we're talking about, but inside the membrane, it'll be nonpolar, in which case the polar regions of the molecule will be tucked inside, and then the nonpolar regions will be on the outside of the protein. Any question about any of that? All right, so amino acids are linked together to make the protein, and we say they're linked by a peptide bond. A peptide bond is when the OH comes off of one amino acid and the H comes off of the other amino acid, and the OH is from the carboxyl group, the H is from the amino group, and then this nitrogen combines directly with the carbon, shown here. That bond is actually rare in biological molecules, and it is rare in the universe, meaning nitrogen does not bond very often with carbon. It does occasionally, but I'm just saying it doesn't happen very often, but it does happen all the time in proteins. And if you don't know, a protein is a peptide. And so we call this a peptide bond because we find this bond in proteins. Any question about that? Which, remember, a protein is a peptide. When you're growing the protein, meaning you're adding the amino acids onto the growing protein, you call it a polypeptide, meaning poly, many peptides. And so that's why it's called the peptide bond. So when the protein is not finished growing, meaning amino acids are still being added to it, you should call it a polypeptide. From something greater than 2 amino acids to something to 1 amino acid less than the entire protein. Once you've added the last amino acid, that's when you can call it a protein. But before that, it should be called the polypeptide. You can also call it the name a growing protein, meaning a protein that amino acids are being added to. Any question about any of that? And then when you attach the amino acids together, water forms, and I didn't mention that, so let me just mention that. And that's why it's called the dehydration synthesis, by the way, because you're synthesizing 2 smaller molecules to make a larger molecule, and then dehydration synthesis because water is formed. When we're talking about proteins, proteins have 4 levels to the protein structure. There's the primary level, the secondary level, the tertiary level, and the quaternary level. Let's watch a little video discussing the levels to the protein structure. Did that open? Yeah, there it is, just a little slow.
Marie Glusenkamp-Perez said she's not worried about the border while the flood of fentanyl is killing kids.
They're gone because you're not willing.
Yeah, we have to have this commercial because it's from YouTube, so please just wait. I can't pass that one up.
In this video, we're going to talk about the structure of proteins. And what basically is a protein? A protein is a polymer consistent of many amino acids. So each amino acid represented by the circle is a monomer that forms the protein. Whenever you have many amino acids, it's also called a polypeptide. And the bond that connects each individual amino acid residue is a peptide bond. So let's talk about the individual structure of an amino acid.
All right, let's skip
So
that.
an amino acid,
You can view it again if you want.
we could form a peptide plus water. And so this amide, a covalent bond. The secondary structure.
Oh, here we go.
Now let's talk about the different levels of protein structure. There's 4 levels that you need to know. The primary structure, the secondary structure, the tertiary structure, and finally the quaternary structure. Now the primary structure is simply based on the sequence of the amino acids found in the protein. The sequence determines the shape of the protein. If you replace just one amino acid with another, it will completely change the shape of the protein. So the shape and its function is primarily determined by the sequence of amino acids. The secondary structure describes the localized shape of a protein. And there's 2 of them that you need to be familiar with: the alpha helix and the beta pleated sheet. In the alpha helix, you could see that it's stabilized by hydrogen bonds. The NH group of one amino acid interacts with the carbonyl group of another amino acid. And each turn contains about 3.6 amino acid residues. Now here is a visual illustration of the beta pleated sheet. Just like the alpha helix, it too is stabilized by hydrogen bonds between the carbonyl group of one amino acid and the NH group of another amino acid. Next up, we have the tertiary structure,
All right, hold on.
which represents,
Let me state that he's only talking about the hydrogen bonds in the secondary structure because he's only discussing the beta pleated sheet and the alpha helix. And the hydrogen bond is very important in these two structures. But you have to realize there are other amino acid interactions in the secondary structure, which he did not talk about. And the main reason why he didn't talk about it is because there's a third secondary structure. Let me see if I can just do it. It's called the random coil. And the protein just coils randomly. Something like that. And there can be interaction between this amino acid here and this amino acid here, and then this amino acid here and that amino acid here, and maybe an interaction between this amino acid here and this amino acid there. And these are forces of attraction. Now, obviously, in the amino acids here and here, there would be forces of repulsion, which he didn't even talk about at all. But my point is, in the secondary structure, there is more than just the hydrogen bond. And I'll talk about that in a little bit. And he didn't talk about any of the other interactions because he's only talking about the alpha helix and the beta pleated sheet. Any question about any of that? And another thing he didn't discuss is the forces of repulsion in the alpha helix, which maybe this one would be repulsed from this amino acid, and that's why they're far away from each other. But we'll skip that for now, and I'll talk about it in a little bit. Let's move on back to the tertiary level.
next up, we have the tertiary structure, which represents the three-dimensional complete folding pattern of the protein. And so here's a visual illustration of it. So we could see some areas is just a straight chain. Here we have an alpha helix, and here we have a beta pleated sheet.
And right there would be the
Now, the
random
tertiary
coil.
structure is one individual subunit. When you combine multiple subunits, you create a quaternary structure. So hemoglobin is an example of that. It has 4 individual subunits, 2 alpha subunits and 2 beta subunits. So hemoglobin is a protein with a quaternary structure. And so that's basically it for this video. Hopefully it gave you a basic understanding of proteins and the 4 levels of structure that they have. Thanks for watching.
All right, any questions about that video?
Yeah, wait. So how is the tertiary structure made? Is it the interaction of the R groups? Is that what you said?
The tertiary structure is the interaction of different amino acids from all over the chain of the protein. And I'll talk about that in just a minute. But it differs from the secondary structure. In the secondary structure, the amino acids are interacting, but only the nearby amino acids are interacting. In the tertiary structure, the amino acids are interacting all over the protein. That's the main difference.
Oh,
Well, there's
gotcha.
one
Thank
other I'll
you.
talk about. I'll talk about one other difference when I get to it. All right, so let's go back to the primary structure. The primary structure is simply the sequence of amino acids in the polypeptide chain. So once the amino acids have been put on the protein, and then the protein released, all it has is the primary structure. So initially, that's all the protein has. However, different amino acids in the primary structure of the protein can then start interacting with each other. And then we get the secondary level of the protein structure. And that is where localized neighboring amino acids start interacting with each other. And they can be forces of attraction or forces of repulsion in the secondary structure. And the secondary structure does give the alpha alpha helix, as well as the beta pleated sheet, and then the random coil. There are different types of secondary interactions. There can be hydrogen bonds, and that is one of the main bonds or one of the interactions in the secondary structure. It's probably the number one most important, but there are other interactions. And that would be where one amino acid, the amino group will interact with the carboxyl group of another amino acid, forming a hydrogen bond. However, there can be other forces of attraction. And this right here would be a nonpolar molecule being attracted to a nonpolar molecule, forming the hydrogen bond. But you can also have van der Waals forces where a nonpolar amino acid will want to be around another nonpolar amino acid. And so there can be van der Waals forces between two nonpolar amino acids. And the best way to think about van der Waals forces is your bottle of liquid olive oil. The reason why the olive oil is a liquid is because the nonpolar molecules want to be around other nonpolar molecules, and they are attracted to each other by van der Waals forces. Van der Waals forces are very weak interactions, but they are strong enough so that the olive oil comes together to form a liquid. If there was not that van der Waals forces in the olive oil, it would not be a liquid. You would have gaseous olive oil, which you can get by obviously heating the olive oil. Any question about any of that? And then if you put the olive oil in the refrigerator, you can actually lock in place the molecules of the oil where they're no longer able to move about and be a liquid and break the bond and form another van der Waals attraction with another molecule of oil, in which case the olive oil becomes solid. And if you put your olive oil in the refrigerator, at least part of it will become solid, if not all of it. Any question about any of that? All right. What other forces of attraction are there? Oh yeah, there can be attraction between an amino acid with a positive charge and an amino acid with a negative charge. I haven't talked about that, but let me go back up here. Back. Right here. And that is this hydrogen can come off, no, excuse me, this hydrogen can come off the carboxyl group and then move over to the amino group, in which case the COO will have a negative charge, and then the NH3 now will have a positive charge. Is that clear? And there are some amino acids, I think it's arginine for one, which normally has a positive charge. And there's another amino acid, I can't think of it what it is, that normally has a negative charge, but there's at least one. I know that. And they could be forces of attraction where the, where am I? Where the, let's go here and here because those are two the same color. Where this one is a positive amino acid, and it's a negative, and that will be a force of attraction between the ionic amino acids. Any question about any of that? All right, so those are the forces of attraction. There can also be forces of repulsion, and that would be this amino acid here is being repulsed from this amino acid. This could be a nonpolar amino acid and that a polar amino acid. They don't like being together, and so they will be repulsed from each other. And then this would be what, a positive? No, that would be a negative and that a negative. So that would be repulsed also. And the other repulsion, I think I already mentioned it, and that is a non-polar amino acid with a polar amino acid. They will be repulsed from each other. As well as, oh, I didn't mention this one. And that is if they both have a positive charge, like this one, if it has a positive charge and that also has a positive charge, they will be repulsed from each other. Any question about that? So there's two repulsions and a large number of attractions. There's van der Waals forces, hydrogen bonds, ionic bond, meaning where there's a positive and a negative. So there's three forces of attraction and two forces of repulsion. Any question about the secondary structure? Now remember, the secondary structure only has amino acids which are nearby each other, interacting with each other. If they're far away from each other, it will not be in the secondary structure. Because that's the tertiary structure, where amino acids from all over the protein can interact with other amino acids. Like for here, we have an interaction, force of attraction between this amino acid at the end of the protein and then this amino acid near the start of the protein. So this is a tertiary structure. And you have all of the forces of attraction and repulsion that you had in the secondary structure can happen in the tertiary structure. There is one additional force of attraction that can happen in the tertiary structure that does not happen in the secondary structure, and that is the disulfide bond. Let me draw this out. Well, if I can get paint, come on. Let me try that again. Don't know why that differs when it comes up. In a amino acid that has sulfhydrogen on it, and I know methionine does. So let's make our protein, and we have a methionine. No, excuse me, cysteine is the one I want. Methionine has a sulfhydryl group, but it's especially cysteine, which forms the sulfhydryl group. And then let's go here. Hopefully I can do this well. Oop. And then we have another cysteine here. Which has a sulfhydryl group here. Don't have that in the right place, but it's a little low, but it's all right. Oops, make that HS here. And then I'm going to attach that to the cysteine. Okay, I don't know if I can erase the H, but let me see if I can. Good, I can. the hydrogens can come off, go into solution, and then the sulfur can form a bond with the other sulfur. This is the disulfide bridge between cysteines. This can happen in a protein. This is a force of attraction, and it is a very strong force. An example of this is when you get your hair permed, what they're doing is they're making this sulfide dibridge in your hair between two cysteines. And it makes a very strong link. This is not a hydrogen bond, that is a covalent chemical bond. Any question about any of that? So it's much stronger than a hydrogen bond. This is a force of attraction between two amino acids, usually between two cysteines, that can happen in a protein. That would link this amino acid to that amino acid. And then all of these interactions between the different amino acids give rise to the tertiary structure of the protein, where the polypeptide then forms the three-dimensional structure, folding into a supercoil. Any question about any of that? Now, if the protein only has one polypeptide subunit, that is all the protein can become, the tertiary structure. Most enzymes, if not all enzymes, have only one polypeptide, and so the highest level the protein can reach will be the tertiary structure. But if the protein is formed by more than one polypeptide chain, you then have a quaternary structure. Only some proteins have the quaternary structure, and that's a protein that has more than one polypeptide. Some proteins that have the quaternary structure include hemoglobin, insulin, and antibodies. Hemoglobin and antibodies are made by 4 different polypeptides. And I think the video told you that hemoglobin has 2 of the alpha helix and 2 of the beta helix, or whatever, the beta polypeptide. It doesn't matter. The point is it's made up of 4 subunits, and antibodies are also made up of 4 subunits. I don't remember how insulin is made, but to get the quaternary structure, you have to have the different polypeptide chains come together and start interacting with each other. Like this amino acid to this polypeptide is being attracted to this amino acid of this polypeptide, and it looks like right here, all of these amino acids are attracted to each other, but right there, that amino acid is being repulsed from this amino acid, and this amino acid is being repulsed from that amino acid. You got that? And you have all of these amino acids interacting between the different polypeptide units to make the quaternary structure. So when all of the polypeptides come together and then start interacting with each other, that is when we have the quaternary structure. Only proteins which are made of more than one polypeptide can have a quaternary structure. And I guess I'll mention an enzyme, catalase, for example, only has one polypeptide. So catalase can only go up to the tertiary structure. It has no quaternary structure. Any question about any of that? All right, let's talk a little bit more about proteins. Proteins can be in basically 2 different shapes, a globular protein or a fibrous protein. A globular protein is one that's compact, roughly spherical. Hemoglobin is a globular protein. Most enzymes are globular proteins. And then the fibrous protein is thread-like. Collagen is a thread-like or fibrous protein. Proteins can be conjugated to another molecule. A conjugated protein has a combination of amino acids with other organic or inorganic components. These conjugated proteins are normally named by their non-protein component. Like a glycoprotein is a protein combined with glycogen, and a glycogen is a carbohydrate. So a protein and a carbohydrate, we call it a glycoprotein. And then a lipoprotein is a lipid conjugated to a protein. You can find glycoproteins and lipoproteins both in your blood as well as in the cell membrane. Any question about any of that? All right. When we're talking about proteins, you can have a protein denature. When a protein loses its three-dimensional shape, which can break hydrogen bonds, you can get the protein to denature. Let me state that you can also form hydrogen bonds and get the protein to denature. For example, a protein which is normally in your body at 37 degrees, so it'll form its three-dimensional shape in your body because it's in the correct environment, the correct temperature. If you were to put that protein in the refrigerator, that protein would probably form additional hydrogen bonds that should not be in that protein. That will change the shape in the three-dimensional shape of that protein, causing the protein to denature. When the protein is totally denatured, the protein will no longer function. If the protein is partially denatured, it will not function at its optimal rate, but it will still function, at least a little. When a protein is in the incorrect environment, meaning the incorrect temperature, the incorrect pH, the incorrect salt concentration, the protein will begin to denature. And when it is in an extreme environment, like the real wrong temperature, the real wrong pH, the real wrong salt concentration, the protein will totally denature and will no longer function. Such as, well, actually, your enzymes, if you were to put that in the refrigerator, that enzyme would not function because that enzyme wants to function at 37 degrees, assuming it's not in your skin, meaning it's inside your body. It will want to function at 37. You put it at 4 degrees Celsius in the refrigerator, it's not going to function. It'll be totally denatured. And then if you put it in boiling water, it'll be totally denatured. You put it in boiling water, you break all the hydrogen bonds. You put it in the refrigerator, you're going to form additional hydrogen bonds. Either way, you can get the protein to denature. Because protein denature is just when the protein loses its correct three-dimensional shape, its correct folding pattern. Any question about any of that? All right, any question about proteins? If not, let's move on to nucleic acids. There are two nucleic acids you need to know, DNA, or deoxyribonucleic acid, and RNA, or ribonucleic acid. They consist of monomers. The subunits are called nucleotides. So all nucleic acid are made up of nucleotides. Nucleotides consist of three parts: a 5-carbon pentose sugar, which in DNA is deoxyribose, in RNA is ribose, a phosphate group shown here, and then a nitrogen-containing base shown here. And there's another nitrogen-containing base shown here, meaning this is another nucleotide. Any question about nucleic acids? All right, so nucleotides contain a 5-carbon pentose sugar, a phosphate group, and a nitrogen-containing base, either a purine or a pyrimidine. In DNA, we have 4 bases: adenine or A, guanine or G, cytosine or C, thymine or T. And I don't care how you know them, adenine or A, either way will be fine. Adenine and guanine are purine. Cytosine and thymine are pyrimidines. How you can easily remember in DNA the pyrimidines from the purines is you look to see if the name cytosine has a Y in it, and if cytosine and thymine have a Y in it, if it has a Y in it, then it is a pyrimidine, which also has a Y in it. A purine does not have a Y in it, so anything that doesn't have a Y in it is a purine. Any question about any of that? Now, RNA has 3 of the same bases. They have adenine, guanine, and cytosine, but they do not have thymine. They replace thymine with uracil. How you can remember that uracil is a pyrimidine is just remember that uracil replaces thymine. Thymine has a Y in it, so both thymine and uracil are pyrimidines. Any question about any of that? The bases bind to each other via hydrogen bonding. So in DNA, the A and the T always bind to each other with hydrogen bonds, and the C and the G always bind to each other. And please realize when I say A and the T, I also mean T and A. And then when I say C and G, I also mean G and C. A does not bind to C, and A does not bind to G, and vice versa. Any question about that? So the A and the T and the C and the G are complementary bases. They will hydrogen bond to each other. The A will not hydrogen bond to G or C, and all the other possibilities. Let me blow this up a little bit. As you can see here, oops, too far. Oh, I went way too far. What's going on here? Way too far. Here we go. I thought I hit just one. I don't know what happened. As you can see, the A and the T can hydrogen bond 2 times. The G and the C can hydrogen bond 3 times. And I don't know where I got that shown. Right there, the G and the C, hydrogen bonding 3 times. So the G and the C bond is a little stronger than the A and the T, because 3 is a stronger bond than 2 hydrogen bonds. Any question about any of that? All right, DNA, the sugar is deoxyribose. DNA is double-stranded, meaning there are 2 strands of DNA which come together to form the DNA molecule. At least normally in our cells, DNA is double-stranded. Any question about any of that? Each strand of DNA is held together by the chemical covalent bonds between the sugar and the phosphate. So the sugar and the phosphate hold the backbone of the DNA together, and that's a chemical covalent bond, very strong bond, holding a single strand of DNA together. On the planet Earth, you don't normally break the chemical covalent bond in DNA. Obviously, you could have an enzyme do it, but normally, in even boiling the DNA, you would not break the covalent bond in a single strand of DNA. And it's the sugar phosphate which is holding the DNA together. The nitrogen containing base does not hold the single strand of DNA together. The nitrogen containing base is like a step on DNA, which is a double helix ladder. And like the nitrogen containing bases make the step on the ladder. The two strands of DNA are only held together by hydrogen bonds, and they only happen between the nitrogen containing bases on the step of the ladder. So DNA, when it's in two strands, forms a twisted spiral staircase, like a twisted ladder or staircase, and the nitrogen containing bases form the step on the ladder, and they're held together only by hydrogen bonds. Any question about any of that? All right. RNA is composed of the sugar ribose. RNA is single-stranded. In our cells, our RNA is only single-stranded. RNA is held together by the sugar phosphate backbone. So even in RNA, the single strand molecule is held together by the sugar phosphate linking together, meaning sugar, phosphate, sugar, phosphate. RNA does have the bases, and it has 4 different bases, but remember, thymine is not there. It is replaced by uracil. There are 3 major kinds of RNA. Transfer RNA, also called tRNA, messenger RNA, or mRNA, and ribosomal RNA, or rRNA. All of the different types of RNA, meaning tRNA, messenger RNA, and ribosomal RNA, are important in protein translation. So, what is the difference between tRNA and messenger RNA? Do they have the same sugar, ribose? That one's an easy one. All right, think about that. And they're all single-stranded. They're all held together by the sugar phosphate backbone. They all have the same bases, and that would be A or adenine, G guanine, C cytosine, and then U for uracil. So what's the difference between tRNA and messenger RNA? Anyone? Anyone have a guess? It's simply the sequence of nucleotides in the RNA. Determine whether it's tRNA, messenger RNA, or rRNA. If the sequence codes for tRNA, then we have a tRNA. If the sequence codes for rRNA, then there's rRNA. And there's only a handful of tRNAs, well, about 20 of them, I guess. Let me think about that. Yeah, about 20 of them. And there are maybe half a dozen, maybe less, of rRNA. And There's a whole bunch of messenger RNAs. For each protein, there will be a different messenger RNA. In discussing the nucleic acids, we do need to say that ATP, adenosine triphosphate, falls under the nucleic acid. It is actually a trinucleotide, meaning adenine, adenosine, phosphate, meaning it has 3 phosphate groups attached to the adenine. It does have ribose, adenine, and then 3 phosphates attached to it. The second and third phosphates, let me blow that up, have the squiggly line for the attachment, and that means that this is a high energy bond, this one and that one. If ever the cell wants energy, it can break this bond, and then that will release a lot of energy that the cell can use for its energy needs. If the cell needs more energy than just breaking off this one phosphate, the cell can then break off this phosphate, and it release additional amount of high energy, which could also serve the cell's needs. So ATP is something that the cell uses whenever the cell needs energy. So ATP is called the energy currency of cells, because usually when the cell needs energy, it will obtain that energy from breaking off the third group of hydrogen to release energy. And then the cell can use that energy for its energy needs. ATP is made by dehydration synthesis. So you can combine ADP, adenosine diphosphate, and phosphate. We have an I there for inorganic phosphate. It does take energy to combine that phosphate to make it a triphosphate, and then you form water. And that's going this way, meaning the arrow's going this way. And then you can break ATP, adenosine triphosphate, by adding water, and then that will break off this third phosphate group, which will release a lot of energy, which the cell can use that energy for its energy needs. Any question about any of that? ATP is frequently used by cells for its energy needs, so we call ATP the energy currency of cells. And as you know, when you need to buy something, you will pull out your dollar or your money, and money is a currency. In the old days, it was about the only currency. But nowadays, obviously, we have credit cards and digital currency and all sorts of things. But in the old days, money was the only currency. One of the main reasons for the cell requiring energy is to make a larger biological molecule, meaning it needs to biosynthesize 2 smaller molecules to make a larger molecule. And usually when the cell makes that molecule, it will need energy, and usually it'll get that energy from breaking down ATP. All right, any question about any of that? So let's talk a little bit about hydrogen bonds. What molecules have we discussed in this lecture or the previous lecture that have hydrogen bonds that give the molecule important properties? Nobody's going to guess. We're just going to move on. All right, we're we're just going to move on. And then what would happen to these molecules if all the hydrogen bonds were to all disappear? Well, would life be possible? Let me see if there's anyone there. Okay, there's 9 other people here. You guys, you need to contribute.